Hello everyone, welcome to the MGI webinar series. Today is the first session of MGI webinar series. From this one, the technology based on MGI sequencing platform, including sequencing principles, experimental procedures, product selection, and quality control standards, and so on, will be shared regularly by MGI field application scientist team and marketing team. I'm Simone Wang, the FAS from MGI, and I will be the host of today's webinar. First of all, please allow me to begin with a brief introduction of MGI and BGI Group. BGI Group is one of the world's largest genomics organizations, and there are many divisions of BGI to deliver insights, including 4 non-profit organizations such as BGI Research, BGI Colleague, China National Gene Bank, and GigaScience, and the other 6 industrial branches focus on various directions. Among them, MGI is the one to provide platforms including genetic sequencers, reagents, consumables, and informatics software to all other branches of BGI Group and our worldwide customers. In 2013, BGI Group acquired Complete Genomics, a listed company based in San Jose, California. And after 3 years of integration and re-innovation, on 13th April 2016, MGI was officially established to focus on the R&D of life science equipment and consumables. With continuous innovation and upgrading of sequencing platforms, up to now in the commercial side, more than 1,100 units of sequencers have been delivered to 300 end users in 16 countries. And in order to better meet the needs of customers, MGI has formed a marketing service network on a global scale. MGI headquarters in Shenzhen and has 3 R&D centers and 7 branches worldwide with different roles and functions. In addition to the 3 R&D centers and the 7 branches, we also have 24 service and application supports in various countries and regions around the world, such as USA, UAE, Australia, Hong Kong, China, and so on, and 31 commercial sports like USA, UK, Germany, Thailand, and Japan. They operate locally with local staff under local regulation and serve as the service and application support centers for MGI customers worldwide. Alright, back to today's topic. So today we will focus on the theme of DNBSEQ sequencing technology, including the DNBSEQ single-strand circularization library construction, DNB preparation technology and loading method, CPUS sequencing technology, and standard bioinformatics analysis. Before we start, let me give some housekeeping announcement. So today's webinar will last around 40 minutes. If you have any question, feel free to click the Q&A button below the video player and submit your questions by the table at any moment during the presentation. And after the whole presentation, we will answer all of questions as well as we can. If not, you could contact with us by email. Next, let me turn the microphone over to my colleague, today's speaker, Chizheng Yang.
Hello everyone, thanks for joining this on-demand webinar here. My name is Chizheng Yang, FAS at MGI Tech Limited. Today, we are going to give you a brief introduction to our DNBSEQ sequencing technology. So, before we begin, let me be clear about the purpose of this course. After this course, you will be able to understand MGI single-stranded circular DNA library and how to generate and load our DNA nanoball on our flow cell. And still, we will then CPAS, combinatorial probe anchor synthesis sequencing technology. And in the end, there will be in the basic data analysis workflow. Well, according to these objectives, today's webinar is going to be divided into 4 parts, starting from the library preparation, DNB generation, sequencing, and analysis. We can start with the first part, library preparation. I think most of you might know how next-generation sequencing or massively parallel sequencing work. Let's choose whole genome sequencing as an example. It begins all with fragmented DNA, and that DNA will be done with end repair attaining, adaptive ligation, and PCR. However, compared to the other platforms, MGI's library is a little bit different because after the regular PCR, there is an additional step called circularization. Let me make this more clear. The double-stranded DNA will be denatured by heating into single-stranded because we designed that blue regions into our adapter, which can be complementary paired to our split oligo. So, after adding split oligo, that oligo helps that single-stranded DNA forms into single-stranded circular DNA. That's our final library. So keep in mind, our final library is not double-stranded PCR product, but single-stranded circular DNA library. If we're trying to understand the structure of single-stranded circular DNA library, we will see there is the green region here. That is our insert sites, where we are interested in. And there is the original part here is our barcode sequence. And also, we designed the gray regions here. It's probably used for annealing our sequencing primer and also our barcode primer. So after successfully constructing the single-stranded library, we should start our next part, the DNB generation. Some people might ask us, what does DNB stands for? DNB stands for DNA nullable. And what is DNA nullable? Why are we using DNA nullable? We will see that single-stranded circular DNA library is our final library. You can imagine it. Single-stranded circular DNA is just like a single match here. If you're going to strike a match in dark light, you will see the light of the single match is too weak, and it is hard to detect by our camera system. So because the signal is not high enough, instead of the single-stranded circular library, we use the DNA nanoball. It's not a single match anymore. It seems like a bonfire in the forest, and which can give off intense light. Therefore, the DNA nanoball is such a structure that can be generated from single-stranded library and also used for enhancing the signal during the sequencing. So the question is, how could we generate DNA nanoball? Or is there any advantages after using that? The answer is yes. For generating DNA nanoball, we use the technology called RCA, rolling circle amplification. So, during the library construction part, we designed the regions for annealing RCA enzyme, RCA primer, and we could add RCA enzyme into it. Using the dark red one represents RCA primer. After adding RCA enzyme, the enzyme will help to extend according to the single-stranded library. Just after one circle, you will see that a single-stranded circular library became double-stranded. Be careful, because I want to make it more clear, so I didn't add the second chain in this animation. So it is single-stranded became double-stranded. Because until the enzyme reaches the starting region of the primer, it turns on the second function of the enzyme, displacement. The generated second chain will be displaced and floating in the solution. At the same time, the extension will be continued, and the extension and displacement are happening at the same time. As time goes on, there is going to be more and more DNA chains generated in the solution. After 300 to 500 of the rotation, we get that so-called DNA nanoball. But be careful, it is not a real ball. It is a single-stranded long fragment DNA. It has beginning and its end. It seems like a ball of yarn on a nanometer scale. Why are we using DNA nullable rolling circle amplification instead of regular PCR as a single enhancement method? Because there are following advantages. You will see the RCA reaction is always based on our original template, which means it is a totally linear amplification with low amplification bias. And also, there is no error accumulation. And by using our MGI Easy PCR-free library preparation kit, combined with our MGI sequencing platform, we could achieve over 99% sleep and precision and sensitivity, and over 99% indel precision and sensitivity. Here is a video shows you how rolling circle amplification works. The double strand was denatured into single. After adding splitting oligo, it forms into single-stranded circular, and which can be used for RCA reaction. During RCA, the original template will be amplified 300 to 500 times. That generates the so-called DNA nullable. And all this process can be done in just one single tube. There is no need to have an external instrument or something, or in one single tube. Well, now we have this ready for sequencing basic unit, DNA nullable. And we have to load that DNA nullable on our flow cell. Here is a picture shows you FCL, flow cell large for our MGI DNBSEQ 2000 RS. If we look at the inside of the flow cell, there are billions of tiny dots here. There will be where the DNB is generating locates and well distributed in the flow cell. If we start to load DNA nullable, we will see each glowing point represents just one DNB. They will well separated according to the pattern array, and which brings you a high imaging efficiency and high density of DNB array. Of course, it produced also high sequencing accuracy as well. As I said before, the tiny block dots are called spores, and the distance between two spores called pitch, center to center, is around 715 nanometer. And diameter of one spore is approximately 220 nanometer. And then the spore point has been emanation modified as positively charged. The other places on the flow cell except spore point will be modified with HDMS. Because of HDMS's chemical function, DNB cannot exist as well except spores. Back to our DNBs. After using adjusted RCA reaction and after controlling duration of the RCA reaction, we control the size of DNB at around 220 nanometer to 240 nanometer. It's a little bit larger than our spore point. So we could use that manual or automatic loading equipment for adding the DNBs on our flow cell. The DNBs will first of all centimate by gravity and will attracted to bind the spores, and it will combine with the spores because of the van der Waals force. After all of them, we will pump that special reagents into it for removing that overloaded DNB and start another process called protein embedding. All of these processes together can ensure that DNBs cannot easily move away. Brief review here. We use another technology for DNB loading. First of all, the flow cell is pattern array designed, and we control the size of pitch, 750 nanometer, the diameter of just one spore point, and the size of DNB itself. Still, the flow cell surface was chemically modified, and finally it's guaranteed that DNB can smoothly load it into flow cell. And each DNB has its own one single spore point. That's our ready for sequencing DNB array. Someone would ask us, is there any particular reason that you made DNB array for sequencing? The answer is yes. First of all, the duplication rate is about duplication rate. So some people might know what's duplication rate. Duplicate means that there are more than one reads which are entirely the same from the first base to the last one, but we need just one. So the exact sequence is so-called duplicate. Higher duplication rate brings for sure high waste of throughput. There are many reasons for duplicate, but one of the major reasons is due to optical crosstalking between DNBs. Because we used pattern array, the DNBs is generated not directly on the flow cell, but outside of them. And at the same time, all DNBs are separated according to the pattern array, so which helps eliminate the duplication rate. Without MGI sequencing platform, the duplication rate can be low as 2%. You might know next generation sequencing is always doing one sequencing run with several samples at the same time. Until the sequencing is finished, we could recognize which reads is coming from which sample. It's all according to the barcode or index. Index hopping, or there is another name, index misassignment, means it's hard, but it becomes easily to be run to identify where the reads originally come from. Higher index hopping could bring less accurate data. This issue is quite essential for some applications like gene expression, microbial profiling, or something. Because with our platform, because we used pattern array flow cell with outside generated DNB, and then we load that DNB onto that flow cell, the index hopping was dramatically decreased close to 1 in 10,000%. The data is from the paper that we published. Here's the reference below. Let's come back to our sequencing process. We had already not ready for sequencing DNB array. We have to put that flow cell with DNB into the machine for sequencing. In this step, we use a technique called CPAS. CPAS stands for combinatorial probe anchor synthesis, which was improved from the CPAL technology of complete genomics. It means like a cycle. It contains 3 steps: incorporation, image capture, and cleavage. So, during the sequencing, we'll pump enzyme and with special DNTP into it. That DNTP has a fluorescent group and a blocking group at the 3' end, which means that only one DNTP can be anchored to our DNA nanoball. Through the excitation of fluorescence group, the DNB emits corresponding light. That light will be captured by our camera system and then transferred to IPC for base calling. After the appears 2 steps, we'll pump cleavage reagent for release that 3' end and makes that DNTP ready for next incorporation. So, which means that one cycle can detect just one base. If you are doing the sequencing of, for example, PE150, that cycles will last 300 times. Here's a video shows you that DNA nanoball array, and we use that CPAS for detecting ATCG. After sequencing, we will get FASTQ data. Somebody might have a question about PE sequencing. As we know now, we are using DNA nanoball instead of regular PCR product. But how could we start for PE sequencing? Let's have a look together. First of all, you will see the gray chain here, which is our template, the DNA nanoball. After read 1 sequencing, some parts of the DNA nanoball become double-stranded. And then, before read 2 sequencing, we start another reaction called MDA, multiplex displacement amplification. So, which we will continue to add DNTP and let them extend along the read 1 strand. When it reaches to the beginning of the next copy, the subsequent strand will be displaced, and that is our MDA strand. Afterwards, the read2 sequencing primers will be alleled to the reverse strand and perform the read2 sequencing. There is one advantage of such a PE sequencing method, is that we know that as the numbers of sequencing cycles increased, the intensity of light will gradually become weaker. However, through the MDA reaction, the number of reverse strands can be generated, and which means the signal of the reverse strand is at least equivalent to the forward strand. After PE sequencing, we could figure out how our sequencing strategy looks like. If it's a single-end SE sequencing, we will start with read1 sequencing. For example, if it's SE50, will be 50 base pair for read1. And then we will perform barcode sequencing. Barcode contains 10 base pair. If it's paired end, we will start with read1, for example, PE100, 100 base pair for read1 sequencing, and 100 for read2, and then 10 base pair for the barcode sequencing. Back to our sequencing platforms. There are 3 sequencing platforms available globally. The benchtop size sequencer, MGI 200, the medium high throughput sequencer, MGI 2000, and still the production scale sequencer, MGI T7. And different sequencing platforms are using different color scheme. For example, MGI 2000 and T7 are using 4-color scheme, which means ATCG are labeled with different fluorophore. There are 4 pictures will be saved after each cycle and used for determining 4 different bases. And MGI 200 is using 2-color scheme, which means that, for example, the light of base T can be emitted by laser number 2, base C can be emitted by laser number 1, and A will be emitted by both laser number 1 and 2. G keep stark. So that's what about our sequencing part. About next part is about the analysis. About the analysis, we can divide analysis into 3 steps. The first one is during the sequencing. The UI software inside our sequencer will save the raw image for further analysis. The next is our primary analysis. Base call software is also inside our sequencer will translate the image file into the base number ATCG. And there will be a separate webinar telling you how base calling works. The peer steps can be done inside our sequencer, and afterwards, with combined bioinformatics accelerator MegaBoard, we can get mapping or SNV or indel detection information. In the primary analysis, we generally want to extract net intensity from the original image and then find out what's the basis behind the light. So the primary analysis contains following steps like raw image detection, and for extracting intensity from the image, and to do the single normalization, and then give each basis a score. Secondary analysis includes the splitting of the barcodes. We know that we are using barcodes for multiplexing or pooling more samples together. Net samples will be separated according to a barcode sequence during the secondary analysis. Next is about our FASTQ structure. There are in total 4 rows here. The first row is the header, start with @sign. The second is our read sequence. You'll see here the read sequence start with base T to the end base C. The third row is a pass sign, and the fourth row is the quality score. If we look at the first row more clearly, you'll see it starts with @sign, and the second one, starting with V, represents the flow cell ID. The third, L1, is the number of the NAND, C001R001 is the row and column of FOV. And 000 is which means the number of the reads, and the last one, number 2, means this read is coming from read 1 sequence or read 2. Well, that's what I want to share with you now. And a quick summary, take-home messages about DNBSEQ sequencing technology. So DNBSEQ sequencing technology contains 3 major parts. The DNA nanoball, MGI single-strand circular DNA library will be amplified by using RCA reaction and finally we generate that DNA nanoball. And DNA nanoball will be loaded on our per-10 array flow cell by using a NOT technology and to form that ready for sequencing DNB array. And afterwards, we will put that DNB array into our sequencer and for running a sequencing by using CPAS, combinatorial probe anchor synthesis. All of this together brings you high accuracy data, reduced index hopping, and decreased duplication rate. Well, that's all about today's webinar. Thanks very much for attention. Thank you.